students, myself Om Prakash Katre and you are welcome to Biology Reward. Today we are discussing the chapter the molecular basis of unit test class 12 and topic today is the genetic code or genetic codon. Student, genetic code or genetic codon. Earlier we have learned about the central dogma. So in central dogma we have learned that the DNA it transcribes to form the mRNA and mRNA molecule will translate to the protein. So this phenomenon it is called the central dogma. In mRNA molecule it get translate to the protein. It means in mRNA molecule there are some specific nitrogenous bases and this nitrogenous bases they code for the protein. They code for the amino acid and that amino acid from the long polypeptide chain and that polypeptide chain help for the synthesis of protein. So mRNA molecule is translated to form the protein. So protein formation takes place and protein formation is take place by the amino acid. So protein is the large molecule which is the polymer of the amino acid. So amino acid is a monomer and this amino acid are helping for the formation of the protein. Now in mRNA molecule it has some specific genetic code or they have a specific genetic codon and this specific genetic codon help for formation of amino acid and this amino acid are for the protein. So we have to discuss now the genetic codon. So in genetic codon there is a little bit history regarding genetic codon which is given in our 12th book. So first we are discussing this one. Genetic codon. George Gamow suggested genetic code should be combination of 3 nucleotides to code 20 amino acid. We know now there are the 20 amino acid and this amino acid, 20 amino acid and these are coded by the genetic code. So the 20 amino acid are formed by the genetic code or by the codon. So he said the George Gamow suggested genetic code, the genetic codon should be combination of 3 nucleotides. So there are 3 codes and these 3 codes help for to code or help for synthesis of 20 amino acid. It means the 3 nucleotides code help for formation of 20 amino acid. Second, H.G. Khorana, it developed the chemical method to synthesizing the RNA molecule with the definite combination of the bases. Now another scientist H.G. Khorana said that he developed the chemical method and he said that synthesis of RNA. He develops the chemical method for the synthesis of RNA and he said that synthesis of RNA molecule with defined combination of the bases. So this RNA molecule synthesized, so he found that when RNA molecule is synthesized, there are some specific combination of the bases. This base may be similar type, so it is called homopolymer. Only one base is repeated many times, it's from the polymer, so it is made up of same type of monomer and that is called homopolymer. Similarly, it may be the copolymer. If different type of bases are combined, then they are forming the molecule. So here H.G. Khorana it developed the chemical method to synthesize the RNA molecule, and he said that there is a combination of bases. The bases are similar, it is called homopolymer, and many similar bases are formed. They form the homopolymer, and different type of bases, it's the copolymer also. So it means during the synthesis of RNA, he said that there is a contribution or combination of bases. And third one, Marshall-Nirenberg. Marshall and Nirenberg, cell-free system of protein synthesis finally helped the code to be deciphering. Okay, so deciphering, it means he said that in Marshall and Nirenberg, in cell-free system of protein synthesis, cell-free system of protein synthesis, he said that there are some codon which are synthesized in the protein. It means this codon, they code the particular amino acid. So deciphering means what? Deciphering is some information which is coded that are translated. So information in the form of data, or information in the data figure, or information in the form of bases, it translated into the amino acid or in the protein. So these three scientists' contribution and their work are very important in this topic, genetic code. First, George Gamow said genetic code should be combination of 3 nucleotide bases, and that's from the 20 amino acids. As Khurana said that when he synthesized the molecular RNA, he said that there is a combination of the bases. Bases will same, then it will form the homopolymer, and if they are different bases, they form the copolymer. Similarly, Marshall and Nirenberg's in cell-free system of protein synthesis, they said that there are some specific code, and this specific code are translated to the amino acid or in the protein. Secondly, the sequence. Now we have to go for what is genetic code. So after this overall discovery and observation and experiment of the different scientists of genetic code, now we have to understand what is genetic code. So students, as I have said to you that genetic codon and the history of the genetic codon, different scientists, they said that the genetic codon, it's help for the synthesis of amino acid. Okay, so now we should understand what do you mean by genetic code or genetic codon. What is genetic codon? The sequence of the 3 nitrogenous bases on mRNA code for particular amino acid is called the genetic code. What is genetic code? Sequence of 3 nitrogenous bases on mRNA code for particular amino acid called the genetic codon. First we should understand RNA is a molecule. In RNA there are nitrogenous bases U, uracil, C, cytosine, A, adenine, and G, it is the guanine. So uracil, cytosine, adenine, and guanine, these are the 4 nitrogenous bases in RNA. Now they said that these nitrogenous bases, they arrange on the mRNA. M molecule, it means it is the mRNA molecule. So the sequence of this nitrogenous bases, C, I am arranging C, U, C, A, G, U, C, A, G, 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 A, this type of codon I am writing. So these nitrogenous bases, they arrange on mRNA. So the sequence of 3 nitrogen, this 3 nitrogen, 3, 1, 2, 3, 1, 2, 3, 1, 2, 3. So this sequence of 3 nitrogenous base on mRNA, it synthesize a specific amino acid. This also synthesize another specific amino acid. This also synthesize another amino acid. So this is amino acid 1, 2 and 3. So what happened? What to understand here? These nitrogenous bases are arranged on the mRNA molecule and 3 nitrogenous bases 1, 2, 3. So these 3 nitrogenous bases, they synthesize the particular amino acid. So this is called the genetic codon. So the sequence of 3 nitrogenous bases on mRNA code for particular amino acid called genetic code. So codon is a sequence of 3 nitrogenous bases, and if these 3 nitrogenous bases are on mRNA molecule, they synthesize the particular amino acid, and it is called the genetic codon. This genetic codon, suppose example we can say U, 3 nitrogenous bases U, they form the phenylalanine. This is the phenylalanine. So this phenylalanine is the amino acid, and this amino acid is formed by the U. This means 3 nitrogenous bases U, they synthesize the phenylalanine. Similarly, C, nitrogenous bases, they synthesize the proline, proline. A, 3 nitrogenous bases, they synthesize the lysine, lisine, lysine. And G, the 3 nitrogenous bases, they synthesize the glycine amino acid. So that's what happened. These 3 sequences of nitrogenous bases, and they arrange on mRNA molecule, and they synthesize the particular amino acid. Suppose U synthesize amino acid, it is phenylalanine. C, proline. A, lysine. And G, glycine. So this now it is clear. The sequence of 3 nitrogenous bases on mRNA synthesize particular amino acid, it is called the genetic code. So this is the definition of genetic code. Now this genetic code is having the some characteristics. So we have to discuss characteristics of the genetic code. Characteristic of the genetic code. First one, characteristic we are discussing. Codon or code is triple. Genetic code is triple. Students, as I told you, 20 amino acid. In 20 amino acid, but nitrogenous base we are having U, C, A and G only in RNA. So if each one nitrogenous base synthesize the one amino acid, so there will be only formation of 4 amino acid. But if you will take the double of this one, in such cases, so if each nitrogenous base form the one amino acid, so 4 nitrogenous bases are there, they will form the 4 amino acid. But we should have the 20. So this one single base is not enough. Then if you will see second condition, if these are taken as a double, so bases if you are taking double, suppose U are 4 nitrogenous bases into 4. So in such cases, we will get the 16 amino acid. So here you can see UC, AC, G, and again it is crossed with the UC, AC and G. Then what we are getting? We will get this one. Here to see UC, A and G, UC, A and G, and thus if you will cross here, so you are getting this type 1, 2, 3, 4, 5, 6, 7, total 16. So it will be U, then UC, then UA and UG. So you will get 16 amino acid will be formed if the base we are taking the double. But if we take the base as a triple, in such cases what happens? The base is taken as a triple, so 4 into 4 into 4. So there 16 amino acid will form, but we have the 20 amino acid. So code is not double. Code single it is not, because 4 amino acid are formed. If we are taking the double nitrogenous bases, 16 amino acid are formed. That is not feasible to code the 20 amino acid. So the 4, 4, 4, it means now if I am taking triple, genetic basis is a nitrogenous basis if we are taking triple. So in such cases, we will get the 64 amino acid will be formed. So this 64 codon will be formed. Here 16 codon will be formed and they will code the 16 amino acid. 4 codon will form the 4 amino acid. But if 4 nitrogenous bases are taken triple, so you should understand how triple will come. U, C, A, G. These are the nitrogenous bases. Here also U, C, A and G. Now each nitrogenous base 1, 2, 3 and 4. Each nitrogenous base is having U, C, A and G. So in such cases, each column will be divided by 4. So 1, 2, 3 and 4. So first column C, U, this will be U and this U. Again column U, U and C. Then next U, U and A. And then U, U and G. So for each one we are getting 4, 4, 4 and 4, 16. So 16, 16, 16, 16 and 16. So you will get the 64 codon. And that 64 codon will be enough to code the 20 amino acid. And that's why we used to say a codon is a triple. Suppose example, AUG is code for methionine. So this is the triple codon. GUU is the code for valine. It is also triple codon. So first characteristic of genetic code is the codon is triple. One example, AUG code for methionine and GUU is code for valine. So this type of 64 codon we will get, and that 64 codon are enough to code the 20 amino acid, and so we used to say this codon is a triple. Second characteristic is the codon is unambiguous or specific. What does it mean? Here codon is unambiguous or specific. One codon always code for one amino acid. So one codon, either it is in prokaryotic or eukaryotic, this one codon always code for one amino acid. And this is called codon is unambiguous or specific. Such as if AUG comes, either AUG codon come in prokaryotic or eukaryotic, it is called the methionine. So methionine is formed. So AUG code when come, then it synthesize the methionine. UUU is code for phenylalanine, and we are indicating PHE, phenylalanine. So codon is unambiguous or specific. It means one codon always code for one amino acid. Next characteristic is codon is degenerate. In such cases what we are learning, some code amino acid. First case I told you, one codon code for one amino acid, and that is codon is unambiguous or specific. Second, I am saying the codon is degenerate. Codon is degenerate, it means one amino acid or some amino acid coded by more than one codon. So if one amino acid is coded by more than one codon, or there are different type of many codon which code for one amino acid. So simple definition is when some amino acid they coded by more than one codon. Suppose you see phenylalanine. UUU also synthesize the phenylalanine. UUC also synthesize the phenylalanine. So these two, both codon are different, but they synthesize when the one amino acid, it is phenyl. Similarly, for glycine is coded by the 4 different codon. GGU, it's also code for glycine. GGC, code for the glycine. GGA for the glycine, and GGG. And that's why amino acid is coded by, if amino acid is coded by more than one codon, and it is called codon is degenerated. There are no punctuation. Another third characteristic, next characteristic is there are no punctuations or comma-less codon. So if mRNA molecule is this, so here I am arranging codon. It is nitrogenous base A, C, A, G, T, A, C, and G. So what happened? U, C, A, G, A, C, G. This nitrogenous bases, there is no such type of punctuations or comma. So after 3 nitrogenous bases, because 3 nitrogenous bases code for the particular amino acid, but in between there is no such type of comma. It means, or this type of punctuation is not there. It means the genetic code are comma-less, or there is no punctuation. It is the next character. Codon is non-overlapping. If you will see this codon, UCAGAC, as I told you, codon, 3 nitrogenous bases code the amino acid. So first one, this one is the UCA, 3 nitrogenous bases. This one GAC, and this is not having the pair. But it is doesn't found that this same nitrogenous bases overlap with this one. Okay, so A is participating in this one amino acid, it will not participate in the other amino acid. So codon is non-overlapping. This condition will not occur. Only this condition will occur. The codon do not overlap. So codon is non-overlapping. Another is codon is universal. Bacteria to human being, if there is universal, it means same thing is coded. From bacteria to human being, UUU is synthesized for the phenylalanine. Either this codon present in prokaryotes or eukaryotic, but UUU, it always code for only phenylalanine. And that's we used to say codon is universal. It means one codon will always code the same amino acid. Either it is in prokaryotic or it is in eukaryotic. Another is initiator codon. Why initiator codon is what? If RNA molecule translate to the protein, it means amino acid are formed, then a protein. So the process of protein synthesis is started with the help of this codon. So this codon is called the initiator codon. AUG code or the methionine, it is the initiator codon in prokaryotic. So initiator codon in prokaryotic is AUG. So AUG is an initiator codon. Now this AUG is having the two important or dual role. This AUG, number one, it code for methionine, amino acid is formed. First function, AUG code for amino acid, methionine. And second function of AUG is it acts as an initiator codon. It means protein synthesis process is started by the AUG. Next is termination codon. Termination codon, there are some codon that stop the process of protein synthesis. So they are called stop codon also. And they do not synthesize the amino acid. So they are also known as nonsense codon also. So termination codon, why they are called? Because they stop the process of protein synthesis. So they call stop codon also. And this codon do not synthesize any amino acid, so they are called nonsense codon. Here UGA opal, UAA and UAG. Always remember UGA, UAA and UAG. These are the three codon. They do not synthesize any amino acid, so they are called nonsense codon. And when this codon come, then process of protein synthesis is stopped. So they are called stop codon or termination codon, terminator codon. And last characteristic is collinearity. Collinearity, it means these nitrogenous bases are linearly arranged on the mRNA molecule. So fashion of arrangement of the nitrogenous bases on the mRNA is linear order. So these are the characteristics or feature of genetic codon. Codon is triple. Codon is unambiguous and specific. Always one codon code for one amino acid. Codon degenerated. Many codon code one amino acid. No punctuation, no comma in between this one. Codon is non-overlapping. This type of overlapping of nitrogenous base do not take place. Codon is universal always. UUU, it means one codon code particular amino acid in bacteria to human being. Codon which help for initiation of protein synthesis is called it is initiator codon, and AUG in prokaryotic is a initiation codon. Termination codon, nonsense codon. This nonsense codon, why? Because it do not synthesize any amino acid and it process of protein synthesis is stopped, so it is called stop codon also. And this codon, sequence of 3 nitrogenous bases, they arrange on the mRNA molecule. This is given in our NCERT book only. Now there are some exceptions also which are not given in NCERT book. So in brief we have to understand some exception of the genetic code. In paramecium, UAA and UGA. UAA and UGA. UAA, I told you, UAA, it is the terminal codon. It do not synthesize any amino acid. But in paramecium, this UAA and UGA, both those are the terminal codon in the prokaryotic. But in paramecium, they code for glutamine. So it is exception. Similarly, I told you no overlapping is there, no overlapping, but theta 134 virus, bacteriophagity, here overlapping is optional. In human, mitochondria, AGG and AGA, these are synthesizing some amino acid here. But here in human mitochondria, the AGG and AGA code for arginine act as a stop codon. So they act as a stop codon in mitochondria. Now, many times question come in the examination. Predict the sequence of amino acid. One question we have to do. Predict the sequence of amino acid from given nitrogenous bases. It means this from this nitrogenous bases, those are arranged on mRNA, what type of which one amino acid will be formed. So sequence I am writing here A, U, G. Suppose this is the mRNA molecule. Then U, 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 C, C, C. Okay, and it is the UAA. This is mRNA molecule. Now question it is asked, from this sequence of, predict the sequence of amino acid. Which one amino acid will be formed from this nitrogenous bases? So as we have learned, the 3 nitrogenous bases will form the one amino acid. So AUG AUG, it code for methionine. So first one is called methionine. The second 1, 2, 3 UUU. So the UUU, it is for the phenyl. So it is from the phenylalanine. 1, 2. This UUU again, it will from the phenylalanine. CCC, as I told you, it's for the proline. So it is for the proline. And UAA is a terminal codon. So nothing will form. So in this one, sequence of amino acid will be methionine, then phenylalanine, and proline, and they do not code the anything. So here will be none of this one. So this is the answer of the given nitrogenous bases. So such type of many questions comes in the examination. So overall to see, there are 64 codon. In this chart also you can see the total number of codon are 64 by 4 nitrogenous bases. So that's the codon is the triple codon. And specific codon, code for the some specific amino acid. So we should remember the codon and their synthesizing the